back to Genes with JoJo. Today we'll be exploring what DNA is and how it is made. This is really cool stuff, but first let's do a quick review from our previous episode, What is DNA? DNA is in its most basic sense an instructions manual for all living cells. DNA tells cells how to make more of themselves and how to function while they are living. So now that we have that down, let's go inside DNA to see its structures.
DNA comes in what's called a DNA double helix. It's called this because the helix looks like a spiraling ladder. The DNA double helix has two main parts, which are a phosphate sugar backbone and nitrogenous base pairs. While this sounds complicated, it's really simple when you think about it like a grilled cheese. In a grilled cheese, you have one slice of bread, whatever you like inside the sandwich, and finally another slice of bread. A DNA double helix is just like a grilled cheese sandwich if the bread is the phosphate sugar backbone and the base pairs are the things inside the sandwich. The phosphate sugar backbone bread in this DNA sandwich is pretty straightforward. It is just made up of phosphate and sugar. The base pairs inside of the grilled cheese are a bit more complicated on the other hand. Let's keep going with the sandwich metaphor. In my perfect grilled cheese, I like to have cheese, grilled onion, tomato, and Thousand Island sauce. Before you judge, it is delicious. I highly recommend this type of grilled cheese.
Anyway, just like the inside of a perfect grilled cheese sandwich, in between the phosphate sugar backbone and DNA, there are 4 different types of base pairs.
These 4 nitrogenous base pairs are adenine, thymine, guanine, and cytosine. Inside the DNA double helix, these base pairs will always be arranged by 2s, which makes the helix look like a ladder. In these pairs, adenine will always be with thymine, and cytosine will always be with guanine.
The reason why is a bit too complicated for this video, but if you're curious, it has to do with hydrogen bonds. An easy way to remember the matching of the base pairs A, T, G, and C is that only A and T makes a word, at. We have gone over a lot of theory today. Let's have some fun and make our very own DNA double helixes out of candy.
For this activity, you will need 1 licorice rope, 4 toothpicks, and 2 marshmallows in 4 colors. This bag has pink, green, orange, and yellow, so I first need to get 2 pink, 2 green, 2 orange, and 2 yellow marshmallows. In this experiment, pink marshmallows will represent adenine, green will represent thymine, yellow will be cytosine, and orange will be guanine. Remember, A and T go together, and C and G go together. Now that we have all of this set up, our first step is to make our phosphate sugar backbone. To do this, we will need to cut our licorice rope in half, like this. Next, make 2 adenine and thymine pairs by putting 1 pink adenine marshmallow next to a green thymine marshmallow on a toothpick 2 different times. Here is 1, and I'm working now on my second. Step 3 is just like step 2. Instead of putting adenine with thymine, we will be putting yellow cytosine with orange guanine. Great! Now we have all our base pairs ready to go. For the final step, we are going to put our paired bases into the phosphate sugar backbone licorice. To do this, put all of our toothpicks lined up into the licorice rope, alternating the marshmallows to be AT followed by CG. Finally, put the other end of the licorice phosphate sugar backbone onto the open end of the base pair toothpicks, and voila! You have your very own candy DNA double helix. Who knew genetics could be so delicious? Well, I will leave you to your candy and see you next time on Genes with JoJo.